the USDA Tropical Agriculture Research Station, or TARS, in Mayaguez, Puerto Rico, bananas and plantains are essential crops. The entire national collection is maintained in three principal forms: as mature plants grown in the field, as tissues preserved in liquid nitrogen at a separate facility in Colorado, and as lab-grown plantlets known as in vitro, or tissue culture. To maintain the genetic integrity of in vitro plants, these must be regularly grown out. This process begins in the screenhouse, where they can gradually acclimate to field conditions.
This is a tissue culture of plants in vitro, transferred to this jiffy pot with soil. We have this plant in this condition over 2 weeks. It's very, very, very important to maintain the humidity, okay? And it's very, very important that do put the plant under the less sunlight. Maybe 1 or 2 months, you can see the plant in this size. In this case, the plant is ready to plant into the field.
After acclimation, these young plants are grown at the field site in Isabela. This site holds more than 160 accessions of bananas and plantains, with 2 replicates for every accession. Once established, this field will replace the existing site, which will then be available for new research projects. Field sites are carefully monitored and maintained to ensure optimal growth conditions. Phenotypic data are collected throughout the life of the plants and include characterizations important for fruit production.
We evaluate this plant using this data sheet. We obtain over 40 descriptors and complete it, then transfer all data to our database, especially field verification experiment.
After plants are evaluated and determined to be true to type, they can be reintroduced into tissue culture. These plantlets can be propagated in protected and clean conditions for several years before needing another grow out. Recently, TARS has collaborated with Bioversity International to perform field verifications for the world's largest collection of musa. In vitro plants and their cryopreserved counterparts were sent to Puerto Rico for a side-by-side comparison in the field. Where in vitro source plants were found to be off-type, their cryo-recovered counterparts could be used instead. Cryopreservation turned out to be critically important for ensuring access to stable genetics in banana, while the horticultural, phenotyping, and in vitro techniques at TARS were essential for evaluating and correcting the collection.